We're excited to have you today for our webinar, and we have a really exciting topic on antibody discovery. In the past, we talked a lot about in vitro using display technologies, but today we're shifting the focus to in vivo, which is really the gold standard when you're talking about biological nuance because there is no technology out there that is smarter than your own immune system. In today's session, we'll talk about how you can unlock more insights from immunization data and go from raw B cell repertoire or single cell data to maturation pathways. My name is Monica. I am the head of go-to-market at my laboratories, and I will be your host today. We're also joined by Sam Poslavsky, CEO and co-founder of MyLabs. And we also have Sophia and Maddie, who are monitoring the chat today. So a reminder to everyone, the session will be recorded, and you can also drop your questions or comments directly in the Q&A and the chat, and we will have time at the end to answer them. On behalf of the MyLab team, thank you so much for taking the time to be here. We know everyone is extremely busy doing their own biologics discovery, especially today in a fast-moving therapeutics landscape. So to give an introduction about us, we are My Laboratories, a software company that is based in San Francisco and in Spain. You may know us as the original creators of MiXCR, which is the most widely used tool for immune repertoire analysis. And today we'll be showing our new software, which is a user interface for biologics discovery. We work with many large pharma, biotech companies, CROs, and all of the top research institutes around the world, and our technology has also been cited in thousands of papers and patents. So that is a little bit us. Let's get started, and I'm excited to pass over to Stan, who will talk about today's agenda.
Thank you very much, Monica, and thank you very much, everyone, for joining this webinar. So, as Monica mentioned, this session is about in vivo antibody discovery. We already had a bunch of meetings and masterclasses about in vitro antibody discovery, where you have phage displays or other display technologies to select your high affinity leads. And in contrary, in vitro antibody discovery, when we use in vivo, we are actually relying on the immune system and the immune system's natural affinity maturation process, allowing us to find the high affinity and developable leads. So the standard pipeline for in vivo antibody discovery starts with animal immunization, and depending on the animal, depending on the species that we select, either let's say mice or llamas, there are two different paths in the in vivo antibody discovery. Specifically today, we'll be focusing on mice models, and with mice, the pipeline is very standard. You immunize mice, then in a certain period of time, you sacrifice the animal, you sort with a FACS sorting antigen-specific B cells, do sequencing, and today our focus is industrial antibody discovery, and the de facto standard in industrial antibody discovery is single-cell VDJ sequencing, allowing us to get the natural pairing of heavy light. And once we sequence with single cell technology, we have the data, we generate the data, and it comes to the data analysis, which is the bottleneck that we are going to discuss today. Why it's the bottleneck? Well, very simple. The immune system is complex, single cell data is complex. Finding the leads that are not just high affinity and maturated, but also developable is a tricky thing. So that's our primary focus for today is the analysis of such data. So talking about the single cell, which is the standard that we see adopted in many biotech and biopharma companies doing in vivo antibody discovery. Obviously, single cell is a very nice because it gives us native heavy light pairing information. Yeah, with certain cells, we're also getting antigen-specific B cells, and we have a natural. Well, since we're doing in vivo antibody discovery, we're getting the natural. We are leveraging the natural process of the immune system, natural affinity maturation, which is great actually, and allows us to really get high very very nice leads. But at the same time, in the single cell, we have some struggles how to select those leads from single cell data. Yeah, we have one of the bottlenecks of the single cell. It's actually pretty shallow sequencing. So with single cells, we are able to generate thousands or maybe dozens of thousands of cells, but we are not generating hundreds of thousands of or millions of cells required to really go deep into the maturation pathways, reconstruct semantic implementation trees, etc., etc. And also, one of the things that I will be showing in one of the traps here is that many of the leads that the immune system produces naturally compared to the in vitro, when you have a luxury of designing the screening library, many of the leads that we have produced by our immune system have actually pretty high risk of liabilities. And since at the end of the day, we want to manufacture those antibodies, that exposes a big risk of failure downstream in the discovery or clinical trial process. So yeah, the agenda for today's in vivo masterclass is the following. So we will use a real world data set. So it's a single cell VDG data set from a published study. So where immunization was performed targeting to as well, actually 3 different antigens that we'll discuss later. Yeah, so there is some clonal expansion. There are some extreme developability risks in the data set. And that's a very, very standard situation that we observe among many of our users and customers. So having this data set, we will do a live demo. I will share my screen. I'm already sharing my screen. I will share my screen with the actual software, and we will crack this data set, the single cell data. We'll try to find the best diverse, structurally sound, and actually developable lead panel. Yeah. And at the end of the webinar, please write your questions, or during the webinar, during the session, just write your questions. So we really enjoy discussion, things that important for you. So what we will learn, we'll learn how to achieve structural, or let's say functional diversity of our lead panel. So instead of just relying on the somatic representation trees or like broader sequence clusters, we will reconstruct the paratope sequences using some machine learning models to find the diverse lead panel. We will use the maturation scores, the somatic hypermutation rate in CDRs and frameworks of heavy light chains to, let's say, find the best like high affinity candidates. Yeah, and same time, given the very frequent situation that when you do in silico assessment of the liabilities in your data set, you will find that many of the antibodies produced by the immune system are liable. So we'll navigate this trap, this challenge, to find the leads that are actually good for the downstream manufacturer. So we will use Platforma. That's our software, which is end-to-end discovery solution for your in vivo antibody discovery. With Platforma, you can go from the raw sequence and data, the FASTQ files that you get from Dynamic Genomics, BD Rhapsody, whatever technology you use, annotating and clonotyping with all the proper layers of sequencing and PCR error correction to reconstruct the real clonotypes, which is actually extremely important for in vivo discovery. In vivo discovery, we are relying on somatic hypermutations, and distinguishing somatic hypermutations from sequencing artifacts or PCR errors is super important to reliably identify the maturated leads. That's where gold standard MiXCR comes into the game. So we will do paratope clustering. We will do SHM and maturation analysis to reconstruct broader lineages of our antibodies, predict liabilities and developability properties of our clones, integrate wet lab data. And the luxury of the study that we have is that actually many of the antibodies that we are going to show today were tested in the lab, yeah, with the ELISA assays. Explore the whole space of the antibodies to build the final lead panel. So
Great. So I just launched a poll. What software are you using for antibody discovery? A few of you have already started to answer it. So we're just curious to know, what kinds of tools you're using. So just answer the poll question, and we'll also have more polls further down this today's session.
Yeah, Monica, please interrupt me because I keep forgetting about the polls.
No
Okay.
worries.
So the data set that we are going to use for the live demonstration is a real data set recently published by AstraZeneca. So here's the study. It was this month, actually in February. It was published in Cell Systems. So the source of the data here is murine models. So there were FACS-sorted antigen-specific B cells collected from donor animals. Yeah, the 10x genomics single-cell EDG sequencing was performed on the sorted B cells. Yeah, and 10x genomics is a standard. We always recommend 10x genomics as the most versatile and at the same time high quality sequencing platform. There were three targets, three arms of the immunization, and three arms in the study. So again, three targets. One is FN14, which is antibody drug conjugate target, very standard one, and where it's explicitly extremely important to select developable leads. Yeah, as we all know, ST2, which is autoimmune target, and but basically we will focus in our live demo on those top two. And the challenge here with the single cell data is that it's shallow single cell sequencing depths. Yeah, so we don't have a lot of clonotypes to reconstruct the full lineage. We don't have time points. Sometimes when we have limited amount of material, we even don't have like compartments, different tissues, etc. So as with all in vivo data sets, it's high rate of liability risks. And one of the key things that many people and many companies are doing, selecting like most abundant candidates or candidates that are supported by the most number of cells. It actually doesn't mean that you're selecting the best affinity candidates. So what we will be doing, we will navigate our analysis through main traps in the in vivo lead selection. Yeah, we'll look what does it mean, the top abundant antibody, and what's the problem, yeah, with selecting just most abundant clones, yeah, which may lack CDR maturations, and result in a weak target affinity. Yeah, so other problem is the mutations in the frameworks. Yeah, so also one of the things that we also need to take into account when there are high, because the immune system doesn't care about our manufacturing, downstream manufacturing ability. Yeah, so the mutations may happen in the frameworks and they may destroy the developability. So our goal will to find the moderate abundance, strong family, and select the perfect leads with a perfect balance between the mutations of the CDRs, clean frameworks, rescue candidates, diverse and free from liabilities. So with this in mind, I'm switching to the live presentation, to the live demo of the software. What you see here is Platforma graphical user interface. Yeah, so it's a desktop app which is connected to the very, very powerful backend where actual heavy load calculation is happening. So here you see a project. Specifically, what you see here is the space of all of the antibodies that we have in our data set. That's a UMAP plot. The idea of the UMAP plot is that two antibodies located closer to each other, if they share similar properties, yeah, we can select a group of antibodies here and compare them on the multiple sequence alignment. So as I mentioned, here we have only single cell data. So for each clonotype, we have a heavy chain and the light chain, thanks to 10x genomics, perfect technology. Yeah. And the whole project you see on the left. This is the structure of our in vivo, very standard in vivo campaign. We're starting by ingesting the data, the FASTQ files in this case. So here we're starting with the raw sequence and data FASTQ files. We import the metadata information, and well, in this specific example, there is not much of the metadata. The next step is the clonotyping analysis, which is done using MiXCR clonotyping. That's the block that we have on the platform, which provides you a very, very important quality control metrics. So first of all, for each of the samples, we see that there are some alerts. We can go to the individual sample and check like what is there. Yeah, like cell barcodes used in the resulting groups is just 14%, which means that there are certain problems in the wet lab which we may address, or maybe it's fine. Yeah. Again, I mentioned that in this specific case, we are using, in this specific study, 10x genomics chemistry is used. So it's 5' single cell VDG kit from 10x genomics. But of course, on the platform, we support all other library preparation protocols. So again, let me just mention one more time for in vivo antibody discovery. When we rely on the somatic hypermutations, it's extremely important to correct for sequence and PCR errors to distinguish them from the real somatic hypermutations in the CDRs and frameworks, and that's what's done here. So this block, MiXCR clonotyping, does this job carefully. If we look on our clones, we have 14,000 of clones of clonotypes in the data. That's what I'm looking in the clonotype browser. For each of the clonotypes, again, thanks to single-cell sequencing, we have all of the information about the frameworks of heavy and light chain. We have information of the full length VG region of the heavy and light. As you see, there are many, many columns, a lot of information generated by MiXCR. The critical information that we will need downstream is, for example, somatic mutations, number of somatic mutations in CDRs, number of somatic mutations on the in the frameworks, on the nucleotide level, on the protein level. As you know, there might be synonymous mutations on the nucleotide level, which result in the same amino acid mutation. So it's a different thing, nucleotide mutation and amino acid. And all this information is really important for our downstream selection of the candidates. Looking on the rarefaction curves, for example, we have an obvious picture. We are not sequencing to enough depths. We are collecting just thousands of cells, and that's what we will always see on the rarefaction analysis and other characterization of our repertoires with single cell data. Yeah. We also may look on the biases in the gene pairing. So, for example, V gene families in our data. Like here, we see that in different. So each sample here is different. Although this is the same murine model, but those are different donors, and in different donors, we may see that certain V genes are actually more abundant. So it tells us that there is some bias maybe in our selection, but those are all very standard things. One thing about this specific study is that a certain amount of antibodies were selected in this study and then tested with ELISA assays. And on the platform, we have a really nice opportunity and a really nice feature to import this functional assay data. That's what happened in here in the immunoassay data block. So we may import the ELISA information right into the project and match this ELISA information or whatever metadata we have about our antibodies with the NGS dataset. It's done in a very simple way. So we have just a TSV file with the information that we got from the functional assays. Yeah, like here, the columns that we import, like binding specificity, like response affinity score, which is manually assigned category medium, high, and low. And yeah, once we import this functional assay data, each of our NGS clones that matches the one of the sequences tested in our functional assay will inherit those properties, and downstream we will use it. So before going into how we select leads, yeah, let's go to the antibody TCR leads block. This is the central place where we select our lead panel. So as you see here, this is the table of the leads that we already selected with different properties. On the settings panel, you see settings that we use to prioritize our leads. So before going into the details, let's just do one simple thing. let's assume that we're selecting the candidates based on their abundance, just for example, to see what we will get there. So I cleaned all of the parameters here, and I put the number of 100 clonotypes to select. So we want to pick from the whole data set 100 of our lead candidates. Yeah, and as a main ranking column, let me select supporting cells. Supporting cells, meaning total number of cells that support this clonotype. Supporting cells is our abundance metric because we have single cell sequence. Let me run this block with this configuration to see what we will get. Okay, done. So here are our leads selected just on the abundance. What's important here is that let me show you the properties of those candidates. So, well, we have first of all what we see here, affinity. Affinity is the property coming from our functional assay data. So, actually, what we did here, we selected those clones that were tested in this specific study with the ELISA assay. More or less, that's how it happens very, very often. You pick the best, the top candidates, and you test them, and that's what actually happened in this specific study. It turned out that the candidates that were picked are most abundant one. Yeah, but then let's check out one thing. Let's check out how developable those candidates are. To test the developability in silica, we have a dedicated block, antibody sequence liabilities. So it's a very, very standard and well adopted in the literature set of patterns in the CDRs of the antibodies that we know may cause either high liability risk and non-developability, or there are certain liabilities that we can actually fix. Yeah, with this block, antibody sequence liabilities, you can configure which of the liabilities you include in the analysis. Yeah, and once you run this block for every antibody, you will have this liability assessment. So first of all, you will have like liability summary for heavy chain and for light chain. What are the general liability patterns we have there? The overall liability risk from high, from non, meaning no liabilities, medium, and low. Those are things that we can fix with immunization, with other tweaks on the protein engineering side, and high liability. Monica, I think you need to mute. And high liability antibodies that are just non-fixable, and we know this for sure from the literature. Yeah. So if we return to our antibody TCR release block, then let me just see where we have this column with the liabilities, liability risk. Let's make it visible, and sequence liability summary. Yeah. So now we have liability risk here, and what we see immediately that majority of the antibodies that we selected by their abundance expose high liability risk. And if we look on the liability summary, we will see that many of those things are what is actually considered the downstream as a bad thing, which is non-fixable. So that's the default approach that we get. If we go to the clonotype space, by the way, yeah, where we have the whole space of our antibodies and color our space by the sequence liability risk, yeah, we will see a set picture that the majority of our antibodies in the data set actually expose pretty high liability risk. Yeah, and only a fraction, like 10% in general, don't have like critical liabilities that we can easily fix. Yeah, and that's the reality. So now let's ask question, how we can build the diversified, well, that's one problem, first of all, that's one problem. Another problem that we can see on our lead panel that imagine we can fix liabilities, but then if we select all of the leads that we have here and compare them on the multiple sequence alignment, yeah, we'll actually see that the diversity of our selected leads is not that great. And Sometimes it depends, of course, on depends on the amount of leads that we are selecting and the antigen. But in general, we may have like groups, really groups of the antibodies, which are looks at least on the sequence level more or less exactly the same. Yeah, and we want to avoid this because we don't want to test 100 leads. Only let's say 30 of them are really diverse, and then there are many variants of our. So we want to achieve a high diversity. So those two considerations. Let's also, before we jump into this lead selection, let's also see one more thing. So we have a tool on the platform which is called Graph Maker, which is really powerful instrument allowing to visualize whatever data you have in your project. It's kind of Prism graph pad allowing to visualize and build publication ready plots. And before we go to the actual selection, let's see what we have. Let's say what we have in general in the project, in the data. So one thing I want to look is the let's say the rate of the mutations in the frameworks. Yeah, mutations framework. So amino acid mutations in the frameworks is just the sum of all somatic mutations happened in the frame. And let's, I don't know, for example, and let's see whether we have any correlation with the liabilities that we have. Well, let's change the type of visualization to the just jitter dots to see what we have. Well, first of all, what we see here, yeah, let me reorder them from none to low, medium, high. Well, what we see here that, well, in general, although we have majority of our antibodies exposing high liability risks, but in general, we see that there are more candidates, more antibodies here with the mutations in the framework, for example. Yeah, which tells us it depends on the data set, but on the many data sets and in many examples that we saw, the higher mutations rate we have in the frameworks, there is a higher correlation with the liabilities. Other thing that we can explore before we proceed to the standardized lead selection is is there any, let's say, correlation or pattern if we consider the rate of the somatic mutations in the frameworks versus the rate of somatic mutations in the CDRs. This will help us to navigate and validate the scoring that we're going to use. So let's say on the on the x-axis, let's put the number of mutations in the CDRs, and on the y-axis, let's put the number of mutations in the frameworks, and we have this picture. Yeah, so that's the number of mutations in CDRs, number of mutations in the frameworks, and all of the antibodies that we have in the data set. Yeah, and let's, for example, since in this specific study we have a bunch of the clones that were actually tested with the lysa, and we imported that information from the immunoassay data. Yeah, we can see. Yeah, we can use the affinity column that we imported from our wet lab assay. Yeah, affinity. We can use this affinity to, let's say, first of all, group our clones by the affinity score. And let's highlight only those that were tested. And I'm moving affinity to highlight to 1, 2, 3. Sometimes my computer is slow, especially when I'm sharing my screen. Yeah, select high and mid. Yeah, so in this specific data set, which is FN14 target, we have only high and medium affinity tested in our candidates. Let me also change slightly the coloring to make it nicer. Let's use this color palette. And for those that were not tested in the functional validation, let's use gray color. Yeah. So what we see here, well, first of all, what we see here that in general, antibodies that among those that were tested in the wet lab validation, yeah, they belong to the lower and have high affinity and in general, like good enough. They are in the lower bar of this plot, meaning that those that have higher number of mutations in the framework in general either don't have good affinity or other problems that we don't select them, or we don't or they fail. If we look on the other data sets, let me switch to the data set which is ST2 target. Yeah, here I have the same plot. We will actually have slightly different picture. So, well, here the order is different. CDRs are on the y-axis and frameworks on the x-axis, but the pattern will be more or less same. The high affinity candidates, if you do deeper sequencing, you will see this in a very nice way. The more mutations you have in the frameworks, the less chance that this candidate is good. But now let's return to the selection. So in the selection, then we can use this information to prioritize our candidates, not just by the total abundance, but for example, by the rate of the mutations in the, let me check, by the rate of mutations in the frameworks or CDRs. Yeah, that's what we can do in the lead selection to prioritize candidates. Actually, in the platform, we have a dedicated in vivo score. In vivo score is just the very simple formula. That's a number of mutations in CDRs divided by the total number of mutations in CDRs and frameworks. Yeah, and the higher, the closer to one it is, the more mutations in the CDRs we have. At the same time, less mutations in the frameworks we have compared to the CDRs. So that's the scoring that we typically found works best to select the candidates and prioritize those that have higher maturation in the CDRs and less mutations in the frameworks that may cause problems downstream. Before going to the final lead panel, two essential steps here. One step is clonotype clustering. So clonotype clustering we have to use to also help us prioritize candidates that are more mature. Clonotype clustering is a very simple block, which, well, I mean, it's not very simple under the hood, but in terms of its parameters, it's pretty straightforward. We select the parts, the features of our antibodies that we want to use to group antibodies together. For in vivo antibody discovery, we always select heavy CDR3 and light CDR3. Yeah, we also put the threshold of the identity of the CDR3, heavy and light here, to group our antibodies together. So in the case of in vivo antibody discovery, single cell, when we have CDR3, considering the length, the typical length of the CDR3, the threshold that we recommend is like 80 to 90%. It works best, but obviously you can add one more clonotype clustering block and try it with different parameters to see what works best for your specific data set. Yeah, and this will actually build us the clusters, or let's say the clonal families, the families of our antibodies. Let's click on one cluster. For example, yeah, we have 90 candidates here. That's heavy CDR3 and light CDR3. Yeah, so that's the family in our like family of our antibodies that we have here. That we may think is a real lineage of in the maturation process. Actually, that's not true, because again, this is a trap of single cell sequencing. It's very shallow. So we don't get there enough of information to reconstruct the real lineage. Let me show you in contrast one example. So I have a project where the llama was immunized, and then the data was collected through time points, and both bulk and single cell sequencing was performed. With bulk, you have really deep sequencing, and you have lots of information to reconstruct the true lineage of the maturation. And with single cell, you get just heavy light pair. So we'll have a dedicated webinar to consider the case when you have both bulk and single cell and how you can integrate this data to refine your selection. But that's not very standard because, well, with bulk, you need to do more work. And for such cases, you can reconstruct like the true lineage. You can see the isotype switching. You can see like lots of the information and the true lineage of the maturation, the true pathway of the maturation on the somatic hypermutation tree level. But we have only single cell. We have only single cell. We have 14,000 of cells in this data set. So the clonotype clustering serves as a proxy to assess the lineage. So it's not the real lineage. And when I click this tree structure, it's not a real somatic hypermutation tree. It's impossible to reconstruct somatic hypermutation tree just from single cell data if we don't have like hundreds of thousands of cells, ideally selected through time points, collected through time points. But this is a proxy. So we consider this group of antibodies kind of coming from the same maturation, from the same SHM lineage. And this is very useful because from this, we are getting one property which is crucial for our lead selection, the size, cluster size. The size of the cluster is kind of proxy for us to the like size of the family. And we're saying, and we assume that the larger size of the family, the more, let's say, immune system actually invested more efforts into maturation of this specific family, meaning that those antibodies probably better than others. They worked better than others in the immune system. So that's one source of information crucial for our smart lead selection. The other source of information, but with this, if we use just this, when we will pick candidates from one family with one big family, they will all look similar. We don't have enough backups to select candidates that don't expose liabilities, for example. Yeah. So sorry, we have backups, but we will test one family. So the other step in the rational lead selection is paratope clustering, and in future or structural clustering. So for that we have different capacities. But what we're saying that works nicely in our experience is paratope clustering. Paratope clustering is another block which also does clustering of antibodies, but not based on their sequence, rather based on the predicted paratope sequence based on all of the CDRs. So under the hood, it also groups antibodies together in clusters, but those are compared to like proxy clusters to the somatic implementation tree. Those are like functional clusters with different paratopes. And now we can configure our antibody TCR leads selection process in a rational way. So first of all, in the workflow preset, we can select In-Vivo score. Yeah, and there are two parts here. First part is the filtering. So in the filtering, we can like set the minimal number of, let's say, nucleotide mutations that we want to have in our antibodies, meaning that there is some maturation process. Yeah, we also can add, and that's what I want to add right now, the cluster size as a filter, and assess only the clusters which were like with more than 10 antibodies in cluster, meaning that that's our proxy immune system invested in the maturation of the specific cluster. Next, we are ranking our antibodies by the In-Vivo score, picking the ones that have highest In-Vivo score, more mutations in CDRs, less mutations in the frameworks. And the last ingredient here is diversification, diversified by. So we may use no diversification, or here we can use diversification by the paratope sequence, by the paratope clusters. So with this diversification, the algorithm will first filter the data set and then go one by one through the paratope clusters, yeah, and pick the candidates from each paratope cluster with the highest In-Vivo score. So we run this, and we will have a lead panel which is diverse enough and selected by those maturation criteria. Yeah. So this in general will have higher diversity, or like that's what we see on the multiple sequence alignment. It's actually a very diverse panel. Yeah. And yeah. If we look on the prioritization, yeah, on the ranks of our candidates, yeah, the ones that with the highest in vivo score, we will see that like the top one actually has the highest of the high affinity supported by the ELISA data, at the same time exposing the medium liability risk, which is good. And here we immediately have a backup. Yeah. So that's like the rational way to prioritize those leads. But the ultimate thing that we can apply here is just add the liability risk filter and pick only those antibodies that don't have any high liabilities, any critical liabilities in their sequence. So running this, we will actually get very. Let's run it. Let's run it. So running this will have, first of all, less than requested 100 candidates, just 84, meaning that there is no more in this data set satisfying these filters. Yeah, but those candidates we know, like we did our best to prioritize those candidates based on their maturation and based on their paratope sequence and diversification that we can achieve max. So at least with these candidates, we know as much as we can from in silico liability prediction that they are developable. If we look on the affinity column coming from our assay validation, we still see that many of them are actually supported already by our like wet lab validation with a high affinity scores, which is very, very nice. Obviously, we didn't test in this specific study, scientists didn't test all of the thousands of antibodies. They picked like 20 antibodies or like for this target, I think it's 18, 12 antibody candidates that were picked in this study and tested with ELISA. We have 12 of them here. And by the way, if we compare the candidates that were selected, there is some diversity, but there are some similarities. There are a couple of the candidates which are very similar. And in our lead selection, we capture all of them with a high affinity. Yeah, we have immediate backup candidates, and we know that they don't have high liability risks. So this is the way how we can, and we squeeze everything from the single cell data set. So basically 84 is the maximum that we can squeeze here. So I think I'm taking a lot of time already. I can speak a lot, and I already see we have some questions. So let me go from this. This is the final. This is our diversified developable lead panel selected with the rational approach that I just described.
Okay,
Yeah.
great. So let's move into our second poll question, which
Mhm.
I will launch right now. So you just saw a little bit about Platforma. We're curious to hear if you're looking to replace your current setup, if you're actively looking at new options, if you're just exploring, or if you're happy with what you have.
And while you're answering this question, I just wanted to mention that we are working a lot to push for best antibody selection approaches on our platform, both for in vivo and in vitro. And today I showed how we can use paratope clustering to select and diversify our lead panel and sequence-based liabilities to early de-risk this panel. But things that are in our roadmap to even further advance this approach are structural liabilities. Sometimes, or well, in some significant portion of cases, we may flag a sequence liability, while when we look on the structure of the antibody, this sequence liability may not be that dramatic, for example. So if we look the structure, and we are bringing structural modeling and structural liabilities, 3D structure prediction and visualizations of 3D structures, of course, on the platform in Q1 and early Q2. Yeah, and this will improve significantly the rational lead selection that we have here. And for model animals, we are also bringing the ability to, let's say, taking the mice antibody, see what will be the best strategy of bringing this mice antibody or alpaca or whatever to the human backbone. So yeah, that's a
Yeah,
roadmap. And
perfect. And speaking about the roadmap, we would also like you to help shape our roadmap. So I'm going to launch the third and final poll question, which is based on this roadmap, which is going to be the most relevant to you. Okay. And while you're doing that, I will let San finish his presentation.
yeah, so with Platforma, what I just showed, you have an end-to-end solution to go from the raw reads to selected leads. You have a great variability in the approaches that you can test on the platform, although we have some standardized and templated recommendations for in vivo discovery and for in vitro discovery with different technologies. Today, I covered single cell. We have also solution for combined bulk plus single cell, or single cell coming from BD Rhapsody, or single cell combined with, let's say, RNA-seq and transcriptomics or site-seq, etc. Yeah, and you can start with Platforma today. Platforma is absolutely free for fundamental academic research. That's part of our company mission to support the fundamental research. It's always and in academia, the antibody discovery that's done in academia is more tricky and more time consuming. So we develop a lot of the features to support this. And for our industry attendees, we have a special offer. So you can get a 7-day free access to our demo projects on our cloud server, although you can run Platforma fully on premise in your infrastructure. But if you want, you can get this 7 days trial access to our demo cloud and 30 days on premise free pilot of the platform.
Great. So now we'll move into Q&A. So we have a lot of questions here. Thank you for dropping them in the chats. So I'm going to start with Kareem. First question is, how is the affinity ranking determined here? Is it theoretical, not empirical, or were they determined through beacon analysis, for example?
Yeah, let me just open that question. So how is Finch ranking right here? Is personal theoretical answer? Yeah, it's theoretical. So, oh, sorry, it's based on the somatic representations that we see in the frameworks and and CDRs, first of all, CDRs. So basically, when we align the sequence of the antibody against the reference, we can see where the somatic mutation is located. And then the affinity, we assume that affinity is related to the maturation. That's why we use In-Vivo. We rely on the natural immune system, that there are more mutations, the more mature sequences, the higher affinity is. That's our proxy. For the functional validation for the ELISA data, we of course do them with ELISA, and specifically in this example, we can map the results of our wet lab testing back on the NGS data set, and actually this closes the loop, and we can further guide our selection from the NGS using this wet lab validation.
Great. And then a message from Alexis, can we add our own liabilities definition?
Yes, it's in the version of the block that's currently released. It's not possible, but that's in our immediate roadmap. So maybe in a week or two, or maximum 3, you will be able to put your kind of patterns of the liabilities in CDRs and frameworks and specify them in the block parameter and use them.
Okay, great. And then from Natalia, what types of animals does Platforma have? Another question is nucleotide mutations were counted in comparison with germline genes. So I'll let you answer those two first.
Mm-hmm. Yeah, so first of all, regarding the species, out of the box, we support, of course, human mice, llamas, alpacas, macacas, chicken, rabbit, rat, sheep. Even we have spalax, which is a very unusual thing. Yeah, and also we have the ability to build a custom reference library. So here we have a MiXCR library builder block where you can just put the FASTA file with the VJ genes, and this will of your novel species, and that's it. Or you can use IMGT reference, and they have more species there.
Okay, great. And then what method is used to build the tree, taking into account SHM of antibodies chain, NJ or ML likelihood?
Yeah.
You use some
So
root for
it's
such tree.
mhm. So it's a very good question. So for when we have enough depths in the data, we can use MyTR SHM trees block, which reconstruct the true lineage, but it will not work out of the box on just shallow single cell data. And here the algorithm takes into account, yes, the semantic representations and the frameworks and the CDRs to group and then reconstruct the lineage. It's maximum parsimony method to reconstruct the tree and to build the clonal families. It's like we actually have a paper in Journal of Immunology published dedicated to this method, and I think we can follow up with the paper uh, post webinar.
Okay, great. So for now, those are all the questions that I see. But we will send out the recording afterwards. And if you have any other follow-ups, I'm available, Stan's available. Just feel free to email us, and we'll get those questions answered. So thank you all for being an incredible participant today, and have a great rest of your day.
Thank you very much.